Yeah, so I'm happy to introduce our next invited speaker, Karina Sand. She's in Copenhagen at the Globe Institute in the geogenetics department, correct? I only met Karina very recently, just a few months ago, when we were both teaching at a class of Nichols in Copenhagen, and was really blown away by the work on sort of how DNA sticks to sediments. And I hope you're going to show some of this today. Great, that's super. So she's a geologist and a chemist, which I think is a quite different background than certainly myself and many of the other people in the room. And so I'm really looking forward to your talk. Thank you.
Thank you for the introduction and for the invitation to this meeting. It's super nice to get people with different backgrounds together in this very diverse field. The movie you just saw, that was 60 base pair double-stranded DNA moving on a mineral surface. And that's kind of the level I'm really interested in, to understand how DNA is bound and preserved and retained on mineral surfaces. So I'm going to talk about taphonomy, the how, why, and ifs. This is me many years ago, a geologist. I was doing a lot of field work in Greenland. It was wonderful. No humans, no organics, no vegetation, just me and my hammer and rocks, really. But then I got more interested in the interface between rocks and life, and how mineral surfaces really plays a role in many forms of life. And did a PhD in chemistry and so on and so forth. And then I got interested in environmental DNA. And from my point of view, I'm very interested in preservation of DNA on mineral surface or any surface. So there's extracellular DNA. Every organism extrudes DNA. It can come into the aqueous environment where we know it degrades within a few weeks. But if it's preserved on a mineral surface or another surface, it has potential to survive across time and also across space. So I would like to talk a bit about provenance, because minerals move. Many minerals, they derive from mountains, travel down, wind, rivers, into the ocean before they finally settle. So the minerals have a travel background. And then there's DNA. There can be a settling particle here at the deep ocean interacting with local DNA. But that particle could also have been traveled through rivers, picking up DNA it met all the way up here. So if the mineral carry a history of its travel path, of the DNA it met on way, really depends on the mineralogy of that particle and of the interfacial geochemistry. And I'm going to talk a bit about that, because we can have a scenario where we have, especially with clays, because they pick up whatever they find en route, and it's hard for the DNA to desorb that again. Whereas other minerals, that's maybe not so much of an issue. So there's a huge variation of minerals and a huge variation of mineralogy, and that mineralogy depend on how the DNA is retained. So here again, you see this 60 base pair double-stranded DNA interacting with a mica surface. Mica, that's a clay basal plane. And now we have DNA interacting. The only way we can see DNA now is because we have a nickel chloride solution. Nickel has a plus charge, highly charged dense, so the DNA is retained on the surface, because the mica is negatively charged, and so is the DNA. If we remove the nickel from the solution, we can't see the DNA, because now it's super poorly adsorbed. And the tool I use here is atomic force microscopy. And you can see the DNA is interacting with each other and also on the mineral's surface. So if we think about DNA, especially in aqueous environment, we can't think of the DNA mineral bound as something static. It's dynamic, and it will respond to changes in the chemistry. So DNA, as I mentioned, is negatively charged in most environmental conditions. And silicates like quartz and flint is negatively charged, carbonates and oxide positively charged. So for DNA to retain on a flint sediment, you would need a positive charged cations like nickel, which we won't have much of in nature, but it can be sodium and it can be calcium and magnesium. Sodium is not super great, but it can kind of do the job vaguely. So if you have a complex traveling, what happens? Let's say we have a flint particle with DNA traveling in a saline environment, and then the environment changes to less saline, the DNA will desorb. But if the DNA is traveling on a carbonate or an oxide, the chemical condition is not super important. So surface charts can be used as a guide to assess if a deposit would contain extracellular adsorbed DNA. But it's not the full story because there's so much more to it in terms of the mineralogy and the surface. So there's a composition, the atomic composition, the surface structure, surface charts, surface area, and active sites of the individual mineral surfaces. And here's some mineral surfaces we kind of often find in our sediments. I omitted the most common one, which is quartz silicate, kind of like dirt. And I'll get back to that one. But here we have calcite, hematite, goethite, they are in oxides, and we have a clay mineral kaolinite here. You see by the color, different chemistry and different structures. And these projections here represent the plane with this figure here. So you can see that the atomic structure varies a lot, and so does the charges from each surface that the DNA would feel. In terms of DNA, in terms of clay minerals, it has a flat surface, but there's also there's also edge sites, and the basal planes are negatively charged, and the edge sites are positively charged. This is kaolinite, a booklet of kaolinite, clay mineral, all of this. So little stacks of kaolinite platelets. And this is the basal plane, negatively charged, and the edge sites are positively charged. So if you were a DNA molecule, it would be easy to stick on the edge sites and only on the basal planes if there would be a charged cation to facilitate the bond. And in terms of adsorption, bulk adsorption, the mineralogy really matters. This is quartz, amphibole, feldspar, normal silicate minerals that are normally the major components of our sediments. And this is how much DNA they can adsorb. Not very much. These green writing here, that's all clay minerals. They adsorb much more. They are less common, but they are smaller. So by mass, it doesn't look like much, but by surface area, they can actually comprise a fair amount of a deposit. They can contain much more DNA than the silicates, and smectite is off the charts because it can intercalate DNA, so it doesn't reach saturation. It can just keep putting in DNA in its interlayer. So we kind of did a little think study on the Cap Copenhagen deposits. So this is the mineralogical composition of the Cap Copenhagen deposits. Most is quartz, then feldspar, tremolite diopside, and then about 7% clays. And these are different clay minerals. So if we look at how much DNA these minerals can retain, at a low concentration of DNA, most of the DNA in the Cap Copenhagen deposit would be associated with clay minerals. And by mass, it's super small amount. And if you had a higher DNA concentration, you would get a bit more input from your silicates also. And then we did these adsorption studies. So we had all these minerals with a known amount of DNA adsorbed. So we tried to extract the DNA we added again. And we did really poorly on the clay. So we also have to think about we are not getting all of our DNA out, especially not from the clay minerals. So it would be nice to talk a bit about extraction procedures as well. And that's also something we're working on. Oh, I do want to say here. So we're really interested in which minerals stabilize DNA and why, because it's not about that positive surface charge. It's also about how many charged sites are there for the DNA to bond to. And if we want to preserve DNA, we need the DNA to be stable on the surface, be bounded to many binding sites. So we use atomic force microscopy. It looks like a little coffee machine, kind of this size, super expensive. And here's Magnus loading the liquid cell. And we can do in situ imaging, video rate, and we can do flow through of different liquids, so we can see how DNA binds as a function of different solutions. So we can stabilize the bond, we can stress the bond if we want to. And we can also do forces. So here we have the AFM tip, the little laser light, and on this gold tip, we can decorate a phosphate group, for instance. So the tip is now simulating a DNA backbone. We can put that in contact with a mineral surface. Then we put the tip close to the surface, it presses in. So now it's in contact, and then we remove the tip again. When we remove the tip again, we can measure the force we need to apply to remove the tip because of the phosphate binding to the mineral. And from that, we kind of get a force curve here. We go in the surface, we press in, we pull back. If there were no force, we would go up here, but because there is force, we have this area here that represents the interaction force, and the work of adhesion is the full area here. So we studied the difference between binding between phosphate on calcite and phosphate on mica. And you can see this is different concentrations and ions. So 10 millimolar and 100 millimolar of sodium chloride, 10 millimolar of magnesium, 100 millimolar of magnesium, and then back to sodium chloride. Calcite responds you get a high force with calcite when you have higher salinity because of the electrical double layer. The electrical double layer decreases when you have a high salinity, so it's easier for the DNA to bind, or it binds stronger. Calcite is positively charged, but for mica, it really needed the magnesium ions to bind strongly. So we can also apply this knowledge when we think about extraction procedures. If we just bind, if I was thinking of your study, Vivian, of the elution buffers. We did some in situ FTIR studies of on DNA binding on goethite, and after 24 hours, or was it even 48 hours, we still see DNA bound to the goethite if we just add an elution buffer. So we may need to think of a different step to decrease that, or to inflate the double layer, to destabilize the bond, to get it off clay minerals or positively charged minerals. But for quartz and silicates, it should be fine with just the 2 hours, because the bond is weak at those conditions. We also do imaging. This is a calcite surface. Calcite is rhombohedral, so we get these edge pits. That's part of the crystal structure. These edge sites are positively charged, and the surface is also in principle positively charged, but it structures water super tightly. So there are three tightly bound water layers on top. So it's hard for DNA to penetrate through that water layer, but it's easier on the edge sites. So here you see the DNA sitting. And this is a circular plasmid DNA, but because of the rhombohedral structure of the calcite, they now look angular. So you look at this as a topographic map. The DNA are the white parts, white is tall, and the dark parts are kind of holes in your topography. So the DNA binds on the edge sides, kind of sits like pearls on a string, and we tested different ions to see if that influenced how the DNA would bind. Sodium, it binds fine. Magnesium, it binds fine. With nickel, there's a bit more charge density available with nickel, so it starts to supercoil and can sit a bit more on the edge side, on the surface sides. Then for mica, negatively charged, we couldn't see any DNA binding in sodium. Sodium is a big ion, 1+, so the charge density is low. Magnesium, smaller, 2+, so it has a higher charge density, and we can see the DNA plasmid sitting super nice. Then with nickel, because of the charge density, we start to see supercoiling. So your solution chemistry really matters if you want DNA retained on silicates. Then we tried different solutions and also wanted to do it liquid. Here we have DNA in air again, sitting in seawater. We see nice plasmids. Then I added 1 millimolar calcium chloride. And now all I see now are these dots. So I thought, okay, is that enough to dissolve the DNA, or is the DNA actually just sitting more in the solution than on the surface? So I dried the sample, and the DNA is still there. But in this scenario, most of it was kind of in the bulk solution. So if you think of degradation. It's easier for DNAs to get to DNA that's in the solution than sitting tight on the surface. So salinity and chemistry of your solution really matters for how the DNA can be preserved across time and space. Calcites, I love calcites, and I wanted to do liquid cell on calcites. You saw before we had nice plasmids. I had a nice plasmid in there. Then I mounted the liquid cell, added a droplet of calcium phosphate, no, sorry, calcium chloride. Calcite is slightly soluble, so it starts interacting with the solution until it becomes in equilibrium, so the surface will start dissolving. So it took me 2 minutes to set this up, and on my first scan, I saw long fragments of DNA. And then I started imaging, and then this terrace here, because it's dissolving, the terrace dissolves, and the DNA kind of follows the terrace around. And then I noticed that the DNA starts to jump over to the next step. And now we have small fragments of DNA sitting on that next step. But the more we dissolve, DNA line up on this step, looking like one long fragment. And that continues as the step keeps dissolving. So what happened here is that the binding to the calcite surface fragments the DNA. And I have a little video here I hope I can play. So the DNA is forced to move because of the dissolution, and then it rebinds again rapidly. And it seems to be stable when it binds to its neighbor, but we saw it moving in fragments, right? So these are small fragments. So in terms of a cave sediment of carbonates, the more that carbonate is stressed, dissolved, the more fragmented the DNA should be in terms of, on the basis of a very model, simple study here. I have a colleague, Colin Freeman, he does molecular dynamic simulations, and we put a DNA molecule close to a calcite surface. And we saw two main interactions as calcium, oxygen from the phosphate group, and then also the amine group binds to oxygen on the carbonate. And this bond here creates a lot of hydrogen bonds, which makes the strand lose hydrogen bonding, which then create a major structural breakdown of the strand when the DNA is forced to move. If it's just one binding event, it will be fragmented a bit, but it will still sit tight. But if it's forced to move, it would just increasingly keep getting fragmented and rebind. So in terms of preservation in sediments, in case of our calcite, it will adsorb regardless the solution composition. And if maintained at equilibrium, it would be stable for quite a long time, we would project. But if there's non-equilibrium conditions, then the DNA will be fragmenting increasingly. DNA mica, and this could be DNA silica, DNA chert flint. It needs charge-dense cations to adsorb. But it can also desorb again without breaking. But there's not a high affinity for adsorption on these surfaces. So I've also been toying with Mikkel about with this, how do we use this information in our interpretations of sedimentary DNA? Can we interpret biodiversity measures across different lithological units? If there's any sedimentologist in here, I apologize. I made these lithologies up. So let's say we have a mica-rich, clay-rich, carbonate-rich section here. Can we interpret changes in biodiversity across these layers? And how do we do that? And also keep in mind that every unit here represent a different depositional environment, different ecological needs. So I think you had a point. We need to talk to a geologist. We need to understand the sedimentology when we interpret our cores, especially. I've seen a tendency for people to sample every 5 centimeters or every 2 centimeters. And I really think we need to be careful with that. We can get much more out of our interpretations if we think about mineralogy. And if we use all of that information that are stored in the depositional and provenance history of our sediments. So here we have another clay mineral chloride. It has positively charged layers and negatively charged layer. And here you have a plasmid molecule, circular fragment, bridging across tightly adsorbed on the positively charged places and and weakly adsorbed over the negatively charged side. So in terms of degradation, we would predict that the DNA would eventually fragment because it's not properly stuck here. So in terms of retention and preservation, and Vivian, it was not super surprising to me, you wouldn't see much DNA from chert into at least extracellular DNA because there's not a lot of affinity for DNA to bind to chert. It will kind of behave like this and then eventually get degraded. But that's only the extracellular fraction I'm talking about now. So the binding strength, surface geochemistry, adsorption capacity, and adsorption dynamics, really all of that matters. And then for ecological interpretations, we should pay attention to lithology when we sample and try to understand that depositional history. And then if we can get more DNA off, that would also help us interpret. We can improve the resolution of our sites, and we can also maybe get the small fraction out and the DNA that are stored tightly, because that could tell a different story, or it could add more to the story. And in any case, it's nice to get all of the data out of the precious samples we have, right? And I just want to talk about environmental DNA also, because in environmental studies, you sample the liquid environment to see who was here. Was there a whale? How's the eels doing, etc. But we also need to be careful about, because sometimes people don't see any signature and it's puzzling. But if we have a high mineral load in these waters, the mineral load will absorb the DNA and bring that DNA with them to the bottom strata, right? So the absence may not mean absence of biodiversity, but just a high mineral load. And also, you can kill any presentation with equations, so don't focus on that. Go to the paper if you're interested. We also have tables, and I didn't want to kill you off with those. But in terms of mineral load, if clay particles travel this path and eventually settle here, and the same with quartz sands we worked here, there's an overrepresentation of benthic species in principle, because you have now a high surface area of minerals present that can absorb a lot of the extracellular DNA there, whereas you don't have a high load of minerals, especially at the deep sea, because it's just clay minerals settling. So you won't maybe collect a lot of the diversity in the water column, but at the benthic species, they would be retained. So in terms of us, what's next, what we are working on, we've now managed, Carlotta, PhD student in my group, we spent a lot of time to get DNA molecules on the tip. And then we put that in contact with mineral surface and can measure the differences in binding between different mineral surfaces. And we want to use that information also to understand retention and kinetics, but also certainly how do we destabilize the bond? How do we get the DNA off the surfaces again? And we also do that in combination with FTIR in situ studies, where we can have a mineral film adsorb DNA in situ over time, and we can dissolve it again, and we can use different solution conditions. And for that, it's a nice setup also to test extraction protocols and to test how to get the highly bound DNA fraction off. And in terms of the forces, it's work in progress, but this is an example of no interaction with the surface. The tip comes in, presses in the surface, tip comes out again, not really a lot of interaction. And this could represent a mica surface, for instance, or quartz. Here we have an iron oxide. We come in and there's so much interaction with the surface. And then we can do these kind of summary plots where we can measure the sea forces over distance. And we can also see all of these little events here. That's a snap-off event, which can represent binding sites, for instance. So we hope to be able to use these force curves also to look into the effect of binding site density on minerals, which will be important to understand how well bonded the DNA is and how to destabilize the bond again. All of this was model samples. We can't see chemistry with the AFM, we can only see topography, so we need to know exactly what we have. So now we're at the stage where we'd like to do more complex sample, and we have a new setup for that in the lab where we can target more complex samples. It won't be a walk in the park, but it will be a combination of atomic force microscopy and fluorescence microscopy and optical microscopy. So here we have AFM image of a biofilm, and we see a lot of EPS, extracellular polymeric substances sitting here. And in terms of DNA preservation, we rarely have, well, we do have minerals that are just the minerals, but usually there would be some kind of adsorption of any organic compound. It can also be draping of other minerals on top of that mineral surface. So to understand the complexity on the mineral surface for DNA binding, how will the DNA bind if there are proteins, if they're biofilm, will they degrade faster? Will they not? And here we have a plastic surface. And then we are also interested in looking into to take individual smaller grains and then see, can we see where the DNA sits here? And if so, how do we destabilize the DNA? So that's kind of what's next for us. And that's Magnus, postdoc a postdoc in my group, and Taro Verma. They have a nice background for targeting these things. So in terms of current challenges and future direction, I think you're spot on with this seminar. We need interdisciplinary educations. And you've had a nice summary slide with all the different disciplines, and each of these little spheres you have represent a lot of disciplines already. So getting us talk together, it's nice here because we're here for common goal, but normally in our everyday lives, and students come in, and we need to be able to reach out more and talk more. And for students, it's hard. So I think we could also think about facilitation. I recently had a PhD course across disciplines for medical students and geologists, and we had a professional facilitator. And within 2 hours, she accommodated this safe atmosphere where people could ask, be honest what they understood and what they didn't understood, which really helped breaking down the barriers of also saying what you have to say without being afraid that it may not be relevant. And then in terms of the future directions for the field, I think the mechanism in complex system is needed at this point, because I think now we, at least in our group, we have a good understanding of the chemistry in pure systems. So we are ready to move on to more complex systems. And Mareike, I'm super impressed of your mission and how your approach to look into the context and the micro texture. It's really a lot of work, and I think it's exactly what we need. So be nice to follow. And I also have some inputs, of course, and stuff we obviously could do together. And then think about taphonomy, how the DNA is preserved, how did it get here? Where was it before? Where was the mineral before? And in terms of extractions, we need those extraction protocols. We all agree on that, but it would also be nice with a set of indicators so we knew which extraction protocol would we need to do on this sediment. And when we have that, we can also start opening up new archives. And I think especially of the iron oxide work we have been doing, I think there is hope for hot and humid regions. We just need to find a way to get the DNA off of those surfaces. And with that, thank you to my awesome group, colleagues, collaborators, and funders. And I failed to add Magnus up here, Magnus Harding. You can find him up on my webpage. He's a geologist. But what I also want to show here is we microbiologists, biospectroscopists, chemists, chemical engineer, biomineral chemist, and environmental science, and bioecologist, biochemist. It's across a wide range within the scientific disciplines, and we really need us all for this. So spot on with this seminar.